Okay, let's do this. Mitosis and meiosis. Who is excited? I know I am. Okay, so just a reminder, prokaryotic cells, eukaryotic cells. So when we're talking about cell division, this is going to be a lot easier. We don't have a nucleus. We don't have all the business separated. It's all just at once. Okay, just a reminder. So multicellular organisms, that's you, use cell division. All of your cells, except for your gametes, are somatic cells. Your gametes, your egg and your sperm, are not somatic cells. They divide by meiosis and they are haploid. Just a reminder. The cell's DNA makes up its genome. We know this from earlier. Again, prokaryote, no nucleus. Eukaryote, there is a nucleus. Again, prokaryote, they have plasmids. They have this bacterial chromosome. It's one big giant thing. Just reminders. The eukaryotic genome. This is definitely a reminder while we are thinking about starting the genetics chapter. You have DNA. A section of that DNA is a gene. That gene is going to be wound around some histones, yes, some chromatin, and it's going to make a chromosome. Then that chromosome is what we're going to see in the cell. So these are the chromosomes, and on there are going to be your genes. On the genes, see, here's the genes, considered a loci. That's its position on the chromosome, right? There is your little loci. This is the loci, and the phenotype for that is going to be, for example, black hair versus blonde hair, brown eyes versus blue eyes. Okay? These are the loci. That is the location of the genes that code for a phenotype. Karyotypes do not line up by size like this. It's just kind of a mess. Homologous chromosomes are two. Your XY, again, your gametes are not considered diploid homologous chromosomes. They're considered something else. Okay, so just know that. And this is trisomy 21. This is Down syndrome. This is a karyotype of somebody who has Down syndrome. Okay, so some genomes have pairs that don't match. For example, the X and the Y do not match. Those are hetero. So basically they're like heterologous pairs, okay? Because you have one X and one Y and they match. Again, reminder, you have DNA. There's a chunk there that is the gene. That's going to be wound around your histones. Okay, that's going to be, again, continual winding heterochromatin until you get a chromosome. Okay, same thing, linker regions, methylation, acetylation to make this region tighter or further apart. Histone DNA complex is called a nucleosome, and the connected DNA strings are called the linker DNA. Chromatin is the fiber. We are not going to, the cell cycle. Okay, reminder, we talked about this in the class in case you weren't there. We have G1. This is interphase. This is just you're chilling, you're growing. Some people are in G0. There's no reason. You have neurons that don't duplicate all the time. Okay, so sometimes it's called G0. But if we're moving around the cycle, you're going to start with G1. Okay? Here it is, again, G1. It's a cycle. It's an ordered series of events. Okay? Interphase. There's three parts. G1, okay? GS and G2. Okay. G1 interphase. Most of the time, it's just a big old blob of chromosomes here, okay? But we're entering G1. So change is not evident. Again, your stomach lining every 12 days. So this is happening every 12 days in your stomach. Sometimes, some neurons, it's not happening. It's just So that's G0. Okay, let's go back. So we're chilling right here. We're going to go into S. Okay, so in S, our DNA is going to replicate. Do you remember how we do this? Yes. Remember we did some PCR. It's going to separate. It's replicated, semi-conservative. Yes, this is all a reminder. So in this S phase, we've just doubled our DNA, this synthesis phase. Okay, so we've made it here. We've doubled our DNA. Look at that. So now we have doubled our DNA, but we also need all the other stuff, right? We need organelles. We got to have that mitochondria. We need all the other cellular organelles, cellular things that we need in there. That's all going to happen here. Our cytoskeleton begins to break down and energy is replenished from this use of synthesis, okay? So we're chilling. We double our DNA. Here, we're making all the things to get ready to replicate. Okay, that's like pre-mitosis, if you will, interphase. Now, we're getting ready for mitosis. Okay? Karyokinesis, also called mitosis, is the first step. The second step is cytokinesis, where we're actually going to divide into two. Okay? Here we go. So here's our interphase. Right, we've doubled our DNA, we've made all the organelles necessary. We're entering prophase, okay? Then we're sort of wandering, pro-metaphase is in between here and sort of wandering towards the middle, getting ready, right? This is when your teacher's like, line up for recess, line up, right? So everybody's getting ready. Metaphase, you're in line, right? Boy, girl, or however you're lined up, like by lunchbox or whatever. Right? Anaphase, you're moving apart, and telophase, you're separated. Okay? Prophase, again, nuclear envelope starts to break down. Right? We're kind of like it's still chaos. The teacher hasn't even said line up yet. Right? Maybe this is like go get your lunchbox or go get your coat on or whatever. Okay? This is the very beginning. We have formed interchromosomes. Prometaphase, your sister chromosomes are attached at this point, okay? You have this mitotic spindle. Metaphase, everybody's lined up at the door, right? We are ready to go to lunch. We are ready to go to recess. We are ready. Sister chromosomes are attached. Anaphase, we're separating. We're going our separate ways. Boys to the boys' bathroom, girls to the girls' bathroom, or like whatever you're doing for recess or whatever you're doing for lunch, you're separating, okay? We're separating, we're about to make two. The cell elongates, and now you get telophase where you're actually starting to form this cleavage furrow. Okay? That's mitosis. Cytokinesis is the cell division completed and the physical separation of two daughter cells. Okay, and remember, in plants, that's going to look a little bit different because we have to have a plant the cell plate here for plants. So that's going to look a little bit different. Okay, internal checkpoints. We can't just willy-nilly be doing this all day long, right? No. We have to be like, okay, is it in order before we move to the next one? So there are some checkpoints here. The first one, the end of G1, before we replicate this DNA, is everything in order? Okay, so death of nearby cells. Everyone's dying. Maybe it's either a good time for us to make some new cells because cells are dying off, or something's terribly wrong. Let's get that fixed before we start replicating damaged cells. Growth hormones, right? Puberty, you gotta just grow. Just grow. There's cell crowding, so maybe we need to turn that off because there's already too many cells. Okay, so G1 external influences are probably the biggest thing that you're going to see in this G1 phase. Okay, this is the first one. Should we go ahead and replicate our DNA? There's also a check to make sure that the DNA that we have is not damaged because we don't want to replicate damaged DNA. If this cell does not meet all of these requirements, it will not be allowed to go into the S phase to replicate the DNA. Sometimes it will fix the damaged DNA. Sometimes it will just chill and wait. Or sometimes it will enter G0 and it'll wait until conditions are better. Okay, so we've replicated our DNA. Now we have synthesized, so we've made it through G1. we've made it through synthesis, okay? And now we have this G2 checkpoint. We have duplicated DNA, we have the organelles that we need, theoretically. So this is going to prevent us entering mitosis if all of these conditions are not met. The most important role of this checkpoint is to ensure that all the chromosomes have been replicated and that replicated DNA is not damaged. So we're not going to go through mitosis if everything is not in order. Do we have the right number of mitochondria? Is the DNA, did it replicate properly? If any of the problems are detected, like there's damaged DNA or there's not enough energy, the cell cycle is halted and the cell attempts to either fix the damaged DNA or fix whatever's wrong. Okay? There's also a checkpoint at the end of metaphase. This determines whether all the sister chromatids are correctly attached. And it will not proceed until all of the spindles are correct. This actually is to prevent non-destruction of the cells. Okay, there are positive regulators that promote movement to the next cells and negative regulators that stop advancement to the next cell. These are expressed in kinases. You do not have to know the name of them. Positive regulators move things to the next cycle. Negative regulators stop. Okay? Negative regulators, positive regulators. A word about cancer. So cancer covers many different diseases. Begins with a mutation that results in a faulty protein that should regulate cell reproduction, but it doesn't. So now you have tumors which result from mutated cells that are growing without this regulation. Okay, so you should have normal homeostasis, and instead you have this massive growth that's unstoppable. Proto-oncogenes are normal genes that code for positive cell cycle regulators. When you don't have that, cells can become cancerous. Okay, tumor suppressor genes. These are exactly what they say. They suppress you making uncontrolled cell replication in the form of tumors. So when they're doing their job and they're activated, they prevent uncontrolled division. Cells with a mutated form of a negative regulator might not be able to stop the cell cycle when there's a problem. This is an example of cervical cancer. The tumor suppressor gene is not working correctly. Okay, and a word about prokaryotes. I feel like we always do this. And a word about bacteria is they typically propagate by binary fusion. They just like clone themselves. Okay, and that's sort of, remember, it's a circle, and they go in and replicate and make a new circle, and then cleavage furrow, and all of a sudden you have two new bacteria, and they do this really fast, right? You put your stuff in the incubator, and by that afternoon, you already have these new E. coli. Okay, that's the end of chapter 10.